been years. All right, this is on. Okay, thank you for coming. It's a long day here. This is number 2 for some of you. Yes, so it's really exciting to get another Moss Landing student out the door. We have people just joining on Zoom. There might be people showing up in a little bit. I want to get to the point. Jaycee has come a long way to give this talk to you instead of taking the easy route and doing an online only talk. She brought herself up here from Southern California to make this day happen, which is, I'm very proud of you for doing that because it means I can get up here. Jaycee is one of our COVID babies. She arrived in fall of 2020, right in the middle of the lockdown, and so that must be an amazing experience where your new life and this grad school that you've heard about being so amazing, and you didn't even get to see it for a while, which was a bummer. So it's really great to see students matriculate and move through the program, having come in during that really rough time. And there's actually a lot exciting things to talk to you about Jaycee right now, which is pretty exciting. She came up here from Southern California. She'd worked with a colleague that I'd never really gotten to work with, but we've kind of collaborated, which is Dave Jacobs. Unfortunately, he's employed by UCLA, and my family doesn't really like UCLA. We're more Trojan fans. And so despite that, Jaycee's application looked fantastic. And she was driven. And you could talk to her about this afterwards if you want. Why she was driven. She wanted to come to Moss Landing. She wanted to get this degree. It was really important for her in setting what she wanted to do. And then she really wanted to get back down to Southern California. And so that kind of defines how Jaycee kind of went through her time here. She was very efficient with her time. She picked up on a microscope technique that didn't require an enormous amount of field work, but was a really critical component in regards to what we don't understand about kelp reproduction, and it followed off of a couple of beer pig theses just before her. So I think she did a great job of kind of looking, reading the room and figuring out what would be an efficient thesis, and she was extremely efficient with her time in regards to getting her proposal done in a timely manner. And maybe it was COVID, maybe you had time to think about all this stuff, and like all the rest of us did, and move your way through, which was great. She did her time here in pretty record time. I mean, she essentially ended up finishing here in a little less than 3 years. She did really well in all of her courses. She got the WAVE Award. She got the Moss Landing Scholar Award. She got the Simpkins Scholarship while she was here. She has presented at the Phycological Society of America, the International Seaweed Symposium. She was a UROC mentor during some of her time here. And then last fall, 2023, she took off. She had gotten everything done except the writing and some of the analysis that she accepted a PhD position at USC with Dr. Sergey Knudsen, doing molecular work in kelp aquaculture, which is really where she wanted to be. She wanted to be in Southern California. She wanted to be in a program that was kind of at the forefront, and she got all that. And so she's in her 3rd year now doing that, and doesn't know, only 3rd year in a dissertation, doesn't have the exact title, but she's doing some aspect, this is my paraphrasing, of molecular analyses of kelp reproduction in ways that can advance aquaculture, which is pretty cool, and Sergey is definitely the person to be doing all that with. So it's been 5 years since she's been here, but she's halfway through her PhD at the same time, which is pretty cool. So I'm pretty excited for that. Like I said, efficiency and time. But given her drive to get out of here so quickly, I don't want to stand up here very much longer anymore. So I really just want to introduce to you Jaycee Lanza with the title of her talk, Effects of Variable Light on Sexual Competition in Kelp Species.
All right, so like Mike's been mentioning, I'm going to try and keep this relatively quick for everybody, but I'm Jaycee, and I'm really excited to talk to you about my thesis today. So as most of us know, kelps are foundation species that support some of the most productive and biodiverse ecosystems in the world. And they do this because they modify the vertical and horizontal structures within the ecosystem that allows for more stability to support more faunal abundance. So one of the ways that they do this is by slowing wave energy. So when really fast water moves through the kelp, it's subjected to that drag that the kelp has, and it comes out the other side slower. This protects our coastlines and also stabilizes the benthic ecosystem. Kelp canopies also reduce, let me get rid of that, also reduce light levels. So the really bright light that hits the surface once it gets through the kelp canopy is reduced by up to 90% by the time it hits the bottom. Kelp uptakes nutrients and carbon, so this helps to clean the water and mitigate the effects of ocean acidification. And because of that, it supports this really high level of biodiversity, from invertebrates to fish to our marine mammals like the little otter up there. Kelp supports so much biodiversity that in just the holdfast, it's been shown that there can be 90 species of invertebrates just in the root-like structure at the bottom. So kelp species vary in morphology, so they support very different species assemblages. So the three kelps you see right here are Egregia menziesi, Nereocystis leucana, and Macrocystis pyrifera. And the biggest difference between all three of these is the size and structure. So Egregia is on the smaller side for a kelp. It's about 5 to 6 feet at a maximum, whereas Nereocystis and Macrocystis are both much larger. They can grow to around 90 feet. And you can see that Egregia and Macrocystis both have this like very bladey structure, so they have lots of that leaf-like blade coming off of it, whereas Nereocystis has a really long stipe and then one big pneumatocyst or float at the top, and then the blades coming off. So the holdfast morphology also changes what kind of faunal abundance they can support. Macrocystis has the largest, kind of most robust holdfast, so it definitely supports the highest amount of faunal abundance down there. And surface texture variability, so the texture of the actual stipe and blades has also been shown to affect the associated species. So that's usually with the epiphytes, an epiphyte being something that grows on top of another organism. So the epiphytic abundance is really dependent on this surface texture and variability. So I did just want to briefly go over the kelp life cycle. I think literally everyone in here probably already knows it, but we're going to talk about it anyways. So the adult kelp, the one that you see out in the ocean, is the diploid sporophyte. And that will have specialized blades or regions of the blades that undergo meiosis and produce zoospores, which will then settle onto the substrate and germinate into either male or female gametophytes. And kelp reproduces sexually, so you will then see egg production from the female and sperm production from the male. And then those will recombine to create fertilization and juvenile sporophytes, and this will be the new generation of kelp. So importantly to note for today, the egg of the kelp, the egg emits the pheromone lamoxarine, and this is not specific to any species. All kelp emit the same pheromone. And this pheromone stimulates both the release of the sperm from the males and the attraction of the sperm to the egg. So because this pheromone is ubiquitous among all species, sexual competition can occur at the gametophyte stage between these kelps. So these two kelps are Pterygophora and Macrocystis. If they release their gametophytes at the same time, one will produce an egg first, and that is Pterygophora. So Pterygophora will then start producing that pheromone, and the sperm from both the Macrocystis male and the Pterygophora male will be attracted by that pheromone to the Pterygophora egg. This will lead to, hopefully, fertilization from the Pterygophora sperm and egg to produce the new generation of Pterygophora. Unfortunately for Macrocystis, then later on, when it's able to produce its egg, the sperm has already gone to the Pterygophora gametophyte, so there's none left to fertilize the Macrocystis. This can lead to really reduced fertility or no fertility at all. So several previous studies have shown that sexual competition between kelp species occurs. Macrocystis and Pterygophora, we just covered. Pterygophora outcompetes Macrocystis. Between Macrocystis and Eisenia is the winner. Between Macrocystis and Nereocystis, Macrocystis is the winner. And between Alaria and Laminaria, Alaria is the winner. So several of these, Howard 2014, Christensen 2018, those are both previous beer pig theses. And another important point is that environmental conditions affect the timing of egg production differently in each species. So for example, if we take this interaction that we were looking at before and raise it from 12 degrees to 16 degrees, we actually see that Macrocystis produces eggs much faster. And this actually can flip that competitive interaction. So this will lead Macrocystis to produce the pheromone and attract the sperm from both species, leading to potential fertilization in Macrocystis and decreased fertilization or no fertilization in Pterygophora. This was a beer pig's thesis as well. So today I really wanted to talk about light, and that's because it's so highly variable within the kelp forest. So like I mentioned before, canopy cover accounts for a lot of differences in light, with that irradiance being reduced to 90% of what it is at the surface at the bottom of the kelp canopy. But light also varies with depth, so you can imagine as you get deeper in the water, you have less light. It varies seasonally, so in the winter we have a shorter day length as well as a different angle of the sun hitting the surface, so we have lower light in the winter, higher light in the summer. Storms cause short-term variation in irradiance that can still affect our gametophytes by covering the sun. And then another big one is suspended particulate matter. So if you have an algal bloom or you have an increase in runoff that stirs up dirt and makes the water less clear, you'll have a really big impact on the light in the kelp forest. So that leads me to my main question for my thesis, which is how does light level impact sexual competition between kelp species? And I've broken this down into two kind of smaller questions that I could really look at under the microscope. So the first one is how does varying light intensity affect the timing of egg production? And then how does varying light intensity affect the number of sporophytes produced? And I looked at these both by themselves and then in culture together to see if the competitive reaction was occurring. So I used 3 of our local kelp species. The first one is Macrocystis pyrifera. That's our giant kelp, and it is the most widely distributed kelp in the world. It grows at a depth of about 6 to 20 meters, and it reproduces all year round. So it has no reproductive window. And it's kind of the dominant kelp from Baja California to central and northern California. And then past that, Nereocystis leucana is the dominant canopy-forming kelp. This one's also subtidal. It grows a little bit shallower, but can go as deep as Macrocystis, about 20 meters. The main difference with this one is it's an annual kelp, so it dies off and reproduces every single year, and it does have a narrow reproductive window. And then finally, I used Egregia menziesi. This one is intertidal, so it can be fully exposed and out of the water, and it can be found subtidally, but it is less common. And Egregia is also perennial, so it does reproduce all year round. So now I want to talk a little bit about my methods. So I collected kelp. 2 of my kelps, Macrocystis and Nereocystis, came from Monterey Bay Seaweeds, just down the way, at the aquaculture facility. And then my Egregia came from Fanshell Beach, which is just south of Monterey. So once I collected the reproductive tissue from these kelps, I brought them back into the lab and made what's called a sporophyll lasagna to stress out the tissue and have it release the spores. So basically clean off all the spores, you layer them between damp paper towels, and then you put that whole thing into a cold room, at 10 degrees Celsius in the dark, overnight. And then you take those pieces of tissue back out, submerge them in the water, and then they release their spores. So then once I had the spore solution, I set up my experiment in the tri-welled Petri dish. So I would start with my species 1, and I would settle that into wells 1 and 3, well 1 being the species 1 monoculture, so that would be just species 1 by itself, and well 3 would eventually become the polyculture with both species. So I'd settle this for 24 hours, and then I would rinse off any spores that didn't attach, and I would settle species 2 24 hours later on top of species 3 or species 1 in well 3 of the polyculture, and also in well 2 by itself as the monoculture. So then after these had all been settled and rinsed, I added my light treatments. So I had a low, medium, and high light, which were 20, 40, and 100 micromoles of photons per meter squared per second. And that really scary unit right there is basically just the amount of light that's reaching a certain area at that time point. And I created these with neutral density filters, so that's just like a colored piece of plastic that you put on top of the Petri dish lid to limit the amount of light that can reach the kelp. I tracked the species in the polyculture by making a little mark on the bottom of the Petri dish and then marking these over time. So on day 1, after I settled species 1, I would go and circle them and see where they landed in the dish. And then day 2, I would settle species 2 and then mark where they were. And then after 2 weeks, I was able to see that they stayed in relatively the same places, so I could track their growth and reproduction. So then in all of our wells, I estimated egg and sporophyte production every single day for 30 days. And I used 2 metrics to kind of assess these things. The first one being the number of days to 50% egg production, and then the second one being maximum sporophyte production per female. So when I say 50% egg production, I'm talking about the amount of days it took for 50% of females to produce an egg. So now on to my results. I want to first talk about my Macrocystis and Nereocystis experiments. So Macrocystis produced eggs faster than Nereocystis under low light. You can see the blue and the yellowish lines, Macrocystis is producing 50% eggs at around 23 or 4 days, and Nereocystis is producing slightly after 25, around 26 days. This was also the light level where both species reproduced the slowest. So Macrocystis also produced eggs faster than Nereocystis under medium light. So we have an increase of a few days for Macrocystis. It's coming in at about 22 to 23, whereas Nereocystis is still hanging out over there just past 25. And finally, Macrocystis also produced eggs faster than Nereocystis in the high light. So this one gets a little bit funkier. Macrocystis monocultures, so Macrocystis by itself, produced eggs around 16 days, with the polyculture lagging slightly behind at around 20 days. And then Nereocystis did increase slightly in timing, so it's around 25 days now in the monoculture. And then that purple line is the polyculture of Nereocystis. It did not actually reach 50% egg production. So now we're going to look at the sporophyte metric. So this would be if fertilization occurred, what did that look like? So both species produced fewer sporophytes under the low light and then produced relatively the same at the medium and high. But what's really interesting about this is that when Nereocystis was cultured with Macrocystis, it produced fewer sporophytes. So there is a competitive interaction occurring, right? So our blue box is the monoculture and our red box is the polyculture. And our bottom here is Nereocystis, so you can see that in that polyculture there were fewer sporophytes produced. So did sexual competition occur between Macrocystis and Nereocystis? We saw all the signs, so likely yes. Our timing of egg production in Macrocystis was faster, and we did see that associated decline in sporophyte production of Nereocystis, so it is likely that sexual competition did occur. So this could have been the result of differences in morphology and physiology that impact their reproduction and sexual competition. And the one main one, we've already kind of talked about this, Macrocystis reproduces year-round, while Nereocystis has that narrow reproductive window. This means that Macrocystis gametophytes are likely adapted to more varying environmental conditions, right? Because they reproduce at multiple different times, so there's different light levels and different temperatures. So they have to reproduce a little bit more quickly. Also, they release sporophylls. They release their spores differently. So Macrocystis has sporophylls at the bottom of the kelp, and the spores are released directly from those into the water column. In Nereocystis, the blades at the top form a sori that then abscisses, which basically means it falls off and sinks through the water column, releasing spores as it goes, so that there's a really high density of Nereocystis spores where that sori tissue settles. So that could indicate that to be a better competitor, we might have to have that really high density of Nereocystis spores in the experiment, which we did not. All right, so now on to our Macrocystis and Egregia data. So Egregia reproduced really quickly, much faster than Macrocystis under low light. So we have this one crossing that 50% line from about 13 to 16 days, and then Macrocystis still over here at about 25 days. Under the medium light, Egregia still reproduced more quickly, but it's coming in at closer to 10 to 14 days, and Macrocystis is like 22. And then at our highlight, Egregia reproduced in 10 days, and Macrocystis reproduced in about 16. So Egregia produced eggs faster than Macrocystis under every single light level by a lot. For our sporophyte data, Egregia produced fewer sporophytes at low light and then was the same at medium and high light. And sporophyte production in Egregia did not decrease in the presence of Macrocystis. Macrocystis monocultures produce more sporophytes with increasing light levels, so they produce the most at 100 and the fewest at 20. And then there was this little competitive reaction that occurred at our highest light levels. So Macrocystis polycultures, that red bar, produced fewer than monocultures under just the high light. So there was competition with Egregia, and Macrocystis was outcompeted. So this means because Egregia reproduced faster, and also decreased the sporophyte production of Macrocystis, that this could have been the result of sexual competition. So that kind of begs the question, if Egregia produced eggs faster at every single light level, why didn't competition always occur? And I think that this is because of the gap between when each species produced their egg. So at our low light level, Egregia produced eggs about 12 to 13 days faster than Macrocystis. At our medium light level, it was about 7 to 10 days faster, and there was no competition. And then at our high light level, it was only 4 to 5 days faster, and we did see that competitive interaction. So this suggests that there could be a narrow window of time in which these species can sexually compete. And we saw that Egregia was the better competitor at high light levels. This could be because it's just exposed to higher light, right? We talked about underneath the Macrocystis canopy, the light's reduced by a lot. Egregia is a lot smaller, and it's in a lot more shallow water, so it's experiencing a lot more light on its gametophytes anyways. Also, Macrocystis is always subtidal, where where Egregia is frequently intertidal. So it's exposed to a lot of stressors. It can be desiccated, it can have changes in salinity, changes in temperature, and of course changes in light. And because of all of these things, it likely has to reproduce really quickly so that it doesn't die due to these changing conditions. So this is just a summary of what we just talked about. I have Macrocystis and Nereocystis at low, medium, and high light levels. Macrocystis outcompeted Nereocystis every time. And then down with our Egregia, at low and medium light levels, we saw that there was no competitive interaction, but at high light levels, Egregia outcompeted Macrocystis. So this shows that not only are these species sexually competing with each other, but light can actually impact the outcome of this sexual competition. So just a few conclusions for you all. Light levels shape competition by affecting reproductive timing. Factors like competition that affect kelp recruitment or the establishment of the new generation of kelp shape the composition of species in the kelp forest. And because these different species support different levels of biodiversity, shifts in the species can affect the entire ecosystem. Therefore, examining something like sexual competition under changing environmental variables can tell us more about niche-based partitioning, which is just where the kelps can exist, and allow us to better predict future kelp distributions based on light. So I'd quickly like to acknowledge everybody here, Mike, Scott, and Max, my committee, all the beer pigs, especially Dan. He was instrumental at helping me learn how to culture and learn how to use the microscope and everything. Zach, who was my undergrad at the time, did a lot of data collection for me and is now a grad student here, which is awesome. Monterey-based Seaweeds for supplying my seaweed, all the staff at MLML for supporting me this whole time, and Simpkins for supporting me financially. Does anyone have any questions?
True to her word.
Quick.
Love it. I have questions.
I'm sure you do.
But I'm going to wait.
Maddie?
Is there a reason you didn't look at Nereo and Egregia? And if you did, do you think there was a competition?
I didn't look at Nereocystis and Egregia because most of the time we don't see them in the same niche space. They don't typically overlap. And I was focused more on macro as just like the ubiquitous kelp species here. If they were to have cultured them together, I think since Nereocystis is so much slower than even Macrocystis that they wouldn't have competed. It would have been completely separate.
Fish boy.
That was fun to see it all put together. My question is about what do we know about the timing of sperm production by the male gametophytes? Trying to understand like why, Macrocystis was not outcompeted by Egregia. Are they also delaying when the male gametophytes are producing sperm?
So I don't know specifically how quickly the sperm responds to the egg production, but my theory about why Macrocystis sperm wasn't attracted to the Egregia egg is that the gametophytes at 10 days are just way too underdeveloped to do anything, the Macrocystis gametophytes. So they were just so immature that they were not even close to being able to produce sperm, and that's why that interaction didn't occur.
Well, now I only have one question.
Nice.
Great talk. I was wondering if you have any data regarding the size of the gametophytes, like specifically comparing Egregia and Macrocystis, like were Egregia gametophytes much quicker at growing and that's why they were able to produce eggs quicker, or were they producing eggs at smaller gametophyte sizes?
That's a really good question, and I haven't done any data analysis on this, but I do have pictures from the polyculture of every experiment. And basically, what I was seeing was that Macrocystis would grow vegetatively, so the cells would reproduce through mitosis, and we'd have a bigger gametophyte before reproduction, whereas Egregia would stay as like 1 to 2 cells maximum and start reproducing. So I think that's partially why we get that really fast timing of egg production.
All right, we have a question online. Can you wave at the camera?
Where's the camera?
Right there. That's
Oh, hi, camera.
PhD advisor would like to have a question. So Sergey is asking, under a warming ocean, do you think Macrocystis might evolve to either be more of an annual as opposed to perennial, or develop warm water tolerances, kind of different extremes to that adaptation?
I could definitely see conditions pushing it to be more of an annual. However, the seasonal die-off part of being an annual, I don't really foresee for Macrocystis. But I do think over time it will become more warm tolerant just because Macrocystis is a little bit more warm tolerant than some of our other kelp species. So it's possible that we could have some thermally resilient kelp in the future.
Fair enough. All right, other questions before I ask mine? All right, so I'm going to follow up on Fishboy's question. So I like this idea of trying to figure out the male because it's never been really tracked. And so that has never been shown. That table you had, by the way, I want your whole introduction because it's much better for doing the sexual competition than the way I explain it. So kudos. But that table you had there where you were talking about the timing, right? And you were demonstrating there you go. That's incredible. Because you're absolutely right. Usually you look at when you look at all the previous work, it's about a 4 to 5 day break between the two. When you push it to 12 to 13, your comment was maybe macro's not ready, the males, which would be fantastic to know, and you'd be the first person ever to really well identify Antheridia. So knock yourself out with that one. The alternative, though, is that macro males are perennial as gametophytes. What about, can you comment about the potential to recharge themselves? So let's just say they were mature and they give up everything after that first round with the Egregia. Could they then recharge and develop more spermatozoids by the time they become reproductive later?
I do think it's possible. So I've never seen that with male because it's really hard to identify, like you said. But I have seen female gametophytes that you can induce egg production, have the eggs basically take the eggs off from the female gametophyte or let them grow into sporophytes and then have them separate, and that female gametophyte can then produce more eggs. So I do think that it is possible.
All right, so my follow-up, you did such a great job of explaining how you tracked everyone for 13 days, or 30 days. Couldn't you track the males after that 12-day period to see if they senesced? Because if they're gone, then you could tell me to be quiet and go away, right? But if they're still there, then there's the possibility. Obviously you weren't looking after that because you're already making sporophytes.
Yeah, you could absolutely look for that.
No, I meant, could you?
Could I, in my data? I mean, 30 days for full cell senescence would be really fast, but theoretically I could.
Yeah, because I mean, salmon do it in what, 24 hours? So, all right, that was my partial question. Here's my other question though. Dan Reed got famous on this, right? Terry interacted with macro in the same kelp forest at the same time, demonstrating that that's why Pterogophora existed, right? That it had this window. You've shown for two other species, that that's not the case. In fact, why does Nereo exist if it's always outcompeted?
Well, it's always outcompeted at the gametophyte stage. It's not necessarily competed when they grow, and they can fit into slightly different niches as they grow as well. So, yeah.
Yeah, so that's what I'm wondering. And Egregia's niche is?
Egregia's niche space is higher up in the intertidal where the light and all the conditions are really rough. So it's likely absolutely outcompeted by when Macrocystis reaches adult size, but not necessarily in the intertidal where the conditions are really rough.
Okay, so this isn't really an existential story for Nereo or Egregia because they have these areas outside. It's really at the boundary where the species overlap.
Yes.
Probably at some kind of transition, either intertidally or with wave exposure.
Yes, or when, gametophytes or spores can theoretically move a pretty long distance. So if you had some crossover in just the spore settlement, that wasn't necessarily the boundaries of each individual kelp forest, that could be a reason why they're not able to establish underneath or within each other.
Wow, that triggered so many more comments.
I was just going to ask, how do you think that, so the really high densities of spores that Nereocystis drops and the potential dormancy of these microscopic stages of Nereocystis plays into, well, they suck at competition, but maybe these other strategies allow them to still survive?
Yeah, that's absolutely true. So one thing I didn't talk about today is the fact that kelp gametophytes can basically seed bank themselves, and theoretically they're doing this in the ocean where they can just hang out and not reproduce and then wait for conditions to be favorable before they reproduce. So I think what Scott's alluding to is the fact that Nereocystis gametophytes could hang out until whatever condition that they don't like is over and then reproduce, and that could definitely be a reason why they're still able to compete and exist as full forests.
So you said that gametophytes can produce multiple eggs. On your Egregia macrocystis graph, Egregia seemingly continued or to have eggs produced as macrocystis was reaching that 50% point. Is there evidence there that now there's sexual competition occurring in macrocystis is winning that competition because the gametophytes are now mature?
That's a really good question. Let me go back to where we can actually see these graphs. So I think potentially a better metric than 50% egg production would have been maximum egg, like the timing to maximum egg production. So you can kind of see these peaks, this is the 100 where they did compete, so they do kind of overlap. But the peaks here, you can see the peak in macrocystis egg production isn't even on the 30-day thing. So I think that when they didn't compete at all, it was because the actual peak in egg production was really far apart. Sorry, did that answer your question?
Sort of. I'm just looking at the overlap there at around 24 days. Is that, are you able to look at that time period specifically and see why is the dip in eggs per female for Egregia because they've inputted so much of their energy into sporophyte development and production, or is it now a competition thing that the macrocystis gametophytes are large enough now and they're mature enough that they're now outcompeting the Egregia ones?
Yes, I totally see what you're asking now. Sorry. But the reason that I had these dips was because they're starting to produce sporophytes, and I didn't count them as one thing. So it wasn't once you produced an egg, you have an egg forever. I counted that as 0 eggs. So I think that that's why the dip is occurring, just because they already moved through that reproductive cycle and not because of a competitive interaction.
Fantastic question, and I think with that, this is the third Moss Landing thesis that's done this, and you guys have all really worked off each other very well in terms of providing this. And in fact, I think you guys are now to the point where you can probably put something together as a review on this, because it really hasn't been done. But I think what's being identified here is the males as being an important component, because if all of a sudden those females are crashing because they got fertilized, and therefore their males have senesced, or not, right? Because we do know they senesce, right? They get very pale and they just start to disappear. I still think it might be in your data though, because if you're circling them and you're identifying them, there might be somebody, maybe an undergrad at USC, who can go through these images and look at the longevity of the males, because I think that would be a fantastic thing, is to actually be able to track. You don't have to track the anthrozoids, just whether they're present or not. So even a bar that showed the timing in which males are present in the culture and then when they're not. Because if for Bennett's answer, if the males for Egregia here, basically crap out at day 17 and there's nothing, then we can just wax poetic all we want, because there's no one around to provide those anthrozoids. Right, so anyways, I think it's great. Additional questions? All right, we're gonna let Jaycee go. Congratulations, well done.
Thank you.
Thank you for coming. Scott, Max, and I are going to take Jaycee into the whale room to finish up the finalities for the exam. There is some food still left over. Be sure, what are we looking at?
We have a lot of food and drinks and everything, so feel free to grab some of that, hang out, or don't.
And we're
whatever.
looking at a USC defense date in 2 years?
Say spring 2028.
Oh, all right. So keep your
Yeah.
eyes out for that one. Again, congratulations, Jaycee.
Thank you.
All right. So Bennett, can you pull up the shades? And then we'll be right back.